Hello, my name is Regine Bergholdt. I'm a medical doctor working at Novo Nordisk. Thank you for this opportunity to share some considerations for trial design and feasibility. Novo Nordisk has experience regarding conducting clinical trials within newly onset type 1, and we have an aim of pursuing the development of disease-modifying therapies. I would therefore like to, from an industry perspective, share some of our learnings and thoughts regarding different scenarios for pivotal trials. The role of C-peptide in clinical development has been discussed during this workshop, and I just want to briefly state again, C-peptide is acknowledged as a surrogate endpoint for preservation of beta cell function. It's an endpoint considered likely to predict clinical benefit, but it is mandatory to demonstrate improvement in meaningful and long-term clinical endpoints to get a drug approved for beta cell preservation. To demonstrate a significant effect on long-term clinical endpoints, however, pose some challenges in a recent onset type 1 population, as I'll try to exemplify in this presentation. So no doubt today clinical benefit needs to be demonstrated beyond changes in C-peptide. And which endpoints could then be in scope? A1C is also a surrogate endpoint, but it's accepted and validated for clinical benefit. However, the baseline level and the variability in newly diagnosed type 1s makes this very challenging, which I'll come back to. Hypoglycemia is of course highly clinical meaningful, and severe hypoglycemia by no doubt is feared and directly matters to the patient. However, severe events are very rare in newly diagnosed type 1 diabetes. I'll get back with some learnings we have had on this. Furthermore, ketoacidosis is also highly meaningful, but again, rare in recently diagnosed. Naturally, discontinuation or pausing of insulin treatment would be important, whereas a reduction in insulin dose is debated. CGM-related endpoints and quality of life measures are not yet validated endpoints. And of course, ultimately, an effect on long-term complications is highly significant and would be highly significant, but not feasible to demonstrate within a reasonable time. I will, as mentioned, share some of our considerations regarding trial design and the needed size of trial. And I have chosen 3 scenarios to use as examples during the presentation. One with A1C and one with hypoglycemic events as primary endpoints, and a third one with C-peptide as primary endpoint and A1C or hypoglycemic events as confirmatory secondary endpoints. And finally, I'll come with some considerations on recruitment and feasibility. The first scenario, what if A1C was to be the primary endpoint? There are some key features which pose a challenge for being able to demonstrate an effect on A1C in this population. We have experience from a phase 2 study in recent onset type 1, where we saw an average A1C at baseline being 7.1%, i.e. not much to drop from. And furthermore, the variation in the change from baseline in our experience with recent onset type 1 is that it's much higher than what is experienced than what's our experience in studies with long duration type 1 diabetes. But now to the effect size. In the phase 2 study mentioned, we saw an improvement in A1C of 0.5% in the actively treated, and a difference of 0.4% versus placebo. However, this difference was insignificant, and this was especially due to the high variation. But if we use this expected treatment difference that we had seen and wanted to design a phase 3 study and power based on this. However, before going to this scenario or the design, there is an element which needs to be acknowledged, and that is the potential change of estimand going from phase 2 to phase 3. In phase 2, a hypothetical strategy estimand can be used, i.e. not in the analysis using the data from subjects that have discontinued. In phase 3, it is, on the other hand, a regulatory requirement to use the treatment policy strategy as estimate in confirmatory trials, following the intention to treat principle. I'll not go into details, but this means something for the expected treatment difference you can power after in phase 3. As you can see at the very bottom here, the example with 0.4% demonstrated in phase 2 using the hypothetical estimand will actually only correspond to 0.34% difference in the treatment policy strategy to be used. But now to how it would look. If a phase 3 trial with A1C as primary endpoint was to be powered after what we saw after 1 year of treatment in phase 2, this corresponds, as mentioned, to an effect size of minus 0.34%. Then to have 90% power to demonstrate a significant effect versus placebo on A1C and C-peptide. I can tell you that C-peptide is not the one driving the sample size. It comes more or less for free in this scenario. Then we would actually need more than 1,300 subjects. A note is that with A1C as primary endpoint, it shouldn't be a treatment target trial naturally, but be on top of standard of care. Of course, if the expected treatment difference, as you can see at the bottom, were much higher and not 0.34%, but for example 0.5 or 0.6%, then numbers can be reduced, as you can see. So no doubt A1C is validated and relevant, but the use in recent onset type 1 population can pose challenges. Average A1C is low at baseline and variation is high. As seen, sample size strongly depend on the expected effect size, and unless this is expected to be very high, numbers may make it almost impossible to run a trial gating for approval in this population. And recruitment is not easy in this population. Another factor is the increased use of CGM and closed loops, which may make it even more challenging to demonstrate an effect on A1C in a trial setting, which I'll come back to. You could speculate if A1C is at all the proper short-term goal for disease-modifying drugs aiming at beta cell preservation. Is it a large difference in A1C within the first 1 or 2 years, which is important, or is it rather, for example, time in range, time in hypo, or other measures? So now the second scenario, what if protection from hypoglycemia was to be the primary endpoint? No doubt that reduction in hypoglycemic events are highly clinically meaningful, and especially severe events, which however are very rare in the period after the diagnosis. As the second bullet says, the clinical relevance of C-peptide is known for the risk of hypos. It's also known that the incidence of hypos increase with the duration of the disease. So an effect of a drug on hypos is expected to increase with both preservation of C-peptide, but also with time on treatment. As mentioned, severe or level 3 hypos are very rare in diagnosis. And as an example, in a 1-year phase 2 trial with more than 300 subjects, we observed only 3 severe level 3 events. So this is an important but not realistic point in this population. A composite endpoint of level 2 and 3 could be suggested, and for the example I will use on the next slide, we have suggested to power for a 25% lower rate of hypoglycemic episodes level 2 and 3 as a clinical difference. And to have 90% power to demonstrate a 25% reduction in level 2 plus 3 hypoglycemic events after 2 years of treatment, 570 subjects would be needed. Due to the assumption that the effect on hypos likely increase with time, and a finding as indicated at the very bottom here of a 16% difference in level 2 plus 3 in a phase 2 trial after 1 year, we have for this example assumed that this effect size would then increase from 16% to 25% after 2 years. But of course, only if this assumption of an increasing effect after 2 years of treatment holds true, then the 570 subjects would be enough to demonstrate a statistical difference that would gate for a potential approval of the drug. As discussed, currently C-peptide is a surrogate endpoint reasonably likely to predict clinical benefit, but it is mandatory to demonstrate that a drug that increases C-peptide levels also leads to improvement in meaningful and long-term clinical outcomes. This third scenario is based on that C-peptide may gate for a conditional approval and a label on preservation of beta cell function. And this if an immunomodulatory agent demonstrated significant C-peptide improvements after, for example, 1 year, and then it's clear that it's needed to additionally show significant effect on clinical outcomes, either in the same trial at a later time point or in a separate trial. So the thoughts behind this scenario would be to power for C-peptide as primary endpoint after 1 year, and power for example, hypos as confirmatory secondary endpoints after longer time. Of course, it should be noted that this would then demand regulatory conditional approval while waiting for the outcome. We have calculated that to have 90% power to demonstrate superiority of active drug versus placebo after 1 year on C-peptide and beta cell preservation in newly diagnosed, which corresponds to the first orange box here, and to also demonstrate 80% power superiority of the active drug versus placebo after 2 years on hypo level 2+3, the second orange box here, we would need in total 650 subjects. So a few last considerations after these scenarios. First one concerns children, which we think needs to be included in pivotal trials for this indication. It's well known that children have a faster decline of C-peptide, and you could therefore expect a higher treatment effect in them. However, the variation in C-peptide is also much higher the younger age. So just wanted to bring a consideration regarding that if, as an example, you studied adults only in phase 2, and you want to calculate the needed sample size for a pivotal phase 3, including both children and adults, then due to this balance between the presumed larger effect in children, but also increased variation, we have pragmatically estimated the effect size to be the same in children as what we observed in adults when calculating the trial size needed. Additionally, recruitment in this population is, as mentioned, highly challenging. We have, for example, experienced that it took more than 18 months to recruit a little more than 300 subjects for a global phase 2 trial that included 17 countries and more than 90 sites. So the feasibility of large phase 3 trials in recent onset type 1 needs to be taken into account, can actually, as we see it, sometimes be prohibitive for conducting industry studies that can fulfill the requirements for regulatory. Lastly, the increased use of CGM is definitely here to stay and needs to be allowed in clinical trials, and CGM-related endpoints may be important endpoints going forward. But an important note is that the use of CGM may, of course, make it even more difficult and potentially non-feasible to conduct trials with a glycemic primary endpoint in this population. If we say that we had expected, for example, a treatment difference of even 0.5% in A1C, and this difference was halved due to the use of CGM, i.e., we could now only expect a difference of, for example, 0.25% in phase 3, then we would need around 2,500 subjects to demonstrate superiority of A1C in a phase 3 trial. And likewise, if an expected treatment difference in hypos were not 25%, as mentioned earlier anymore, but only half of it, then we would also need around 2,500 subjects. And my last slide. And in conclusion, of course, what's clinical meaningful for the patients is of utmost importance. As shown, demonstration of improvement in long-term clinical endpoints can be challenging in recent onset type 1, and the choice of the most appropriate endpoints is key. C-peptide is by no doubt the natural primary assessment of preservation of beta cell function with disease-modifying treatment in this indication. And to summarize again on the glycemic endpoints, A1C is often not feasible, and it can be debated whether it is the aim in this population. Level 3 hypos is not feasible as demonstrated. A composite of level 2+3 may be, but it isn't settled which magnitude of effect constitutes a relevant difference and is required for approval. Furthermore, CGM-related endpoints can be considered going forward, and the use of insulin dose can be considered as well. This was actually my last slide. Thank you very much for your attention.